Hello, my name is Boadu, or I'll be known by Josh for this. I'm going to be talking about Christian ethics in gene editing. So starting off with my presentation, we're going to cover CRISPR-Cas9 in biology and how bacteria use this to incorporate in their immune system. We'll then talk about biotechnologies and how we use Cas proteins in humans, especially in the medical field. From there, we'll talk about sickle cells and how treating that is a good way to use CRISPR-Cas systems. Afterwards, we'll talk about other genetic modifications that we could do with our body with the same processes. Later, we'll talk about image of day because we want to know what is the right and wrong way to use gene editing, more so the right way. And lastly, we'll conclude everything in the end. But firstly, what is CRISPR-Cas? So the whole thing stands for Clustered Regulatory Interspace Short Palindromic Repeats and CRISPR associated. So the CRISPR sequences are short 20 to 50 nucleotide sequences, and the reason why it's short is so that the Cas proteins can incorporate this DNA into their sequence. Now, additionally, the genes that code for the Cas proteins are nearby, and I can't say a specific distance because it's different for each bacteria, but they're nearby so that they could effectively mobilize an immune defense in a quick manner. This all happens within 3 short steps. First, you have adaptation, and there's 2 different ways to do this. There's naive adaptation, where you have Cas1 and Cas2 that capture DNA fragments from a new infection. So after everything is all said and done, they capture the remaining fragments and incorporate it so that next time they'll be able to fight it off better. And that's when primed adaptation comes in. You're able to fight off the immune defense. It's like you're getting chickenpox for the second time. You're not going to be hit as hard, or you might be immune entirely. In the case for bacteria, they're still going to get infected, but they'll be able to fight it off faster, and they'll incorporate more viral DNA into their sequence. Now, for expression, there's not much to say about it. It's simply transcribed the same way any other nucleotide sequence is transcribed. But lastly, we have our interference. So expression transcribes cRNA, which acts as messenger RNA, and this transfers the Cas proteins to the viral sequence. So the cRNA transports Cas proteins. In this case, we'll be talking about Cas9, and essentially they effectively cleave off the viral DNA. If you don't have DNA to transcribe, you can't make viral proteins, and the cell can't be infected, therefore preventing the actual infection from starting. So starting off with Cas9, I just wanted to say there's other Cas proteins, but for this we're going to talk about Cas9 mainly because it is a single multi-domain protein, and the reason medical people chose this is because we're able to mutate it, and it's better than running around with several different proteins in the body and trying to manage that. So Cas9 has the pi domains, and these are mainly to bind to the PAM motifs on the viral sequence. And these target G-rich domains, so 3 G nucleotides or more, and then it'll bind onto it. That's when Rex starts to proofread. Now, Rex 1, 2, and 3 do the exact same thing, and some bacteria even 4 and 5, but overall it reads it like polymerase, but it doesn't do what polymerase does. And afterwards, it organizes the nuke domains so that it can undergo cleavage. Now, if the bacteria is not lined up correctly, it'll move around a bit to make sure it's at the right locations. If not, the protein will try and cleave it and fail, and soon disassociate. Now, let's say they do line everything together. What it'll do is activate the nuke domains. And we have RUBC, which cleaves the CRISPR sequence that we just made, so that messenger RNA. And we have the ACE and H domains that cleave the viral DNA code. And again, when it's cleaved, you can't transcribe anything. So how we use this in biotechnologies, because this is the foundation for all 3 of the ones I'll talk about, and we'll bring up all 3 later in the presentation. So the main one is base editors, and these operate simply by fusing nickase Cas9, and these attach to deaminase enzymes. And the benefit about this is that deaminase has a property that allows it to be hydrolytic, and instead of cutting the entire nucleotide, it's able to remove amino groups and substitute them for other amino groups. So you're able to correct what was mutated without actually breaking the DNA. So you don't need to go through homology-directed repair, and that's good for cells that can't do that. And there's a lot of cells in the body that aren't able to do so. Secondly, we have our rec domains, or not rec. Secondly, we have our prime editors. Sorry about that. So these work a little differently. They have Cas9, but they also use reverse transcriptase. And the benefit about this is that it adds RNA extensions from prime editors to GNR, or from gRNA, to the end of a DNA sequence. So it simply adds it, and the HDR homology-directed repair is able to transcribe it on its own. This is a more natural process, and it's able to fix fringe mutations, which are a lot harder to deal, so those are the cause of diseases like cystic fibrosis and retinitis. Lastly, we have our epigenome editors, and these are a little more temporary, and they're also newer. However, the main function of this, they act as histones. And how to do so is they completely deactivate all the nuke domains, so you only have the rect domains and you only have the pi domains. So you're able to bind to it, make sure you bind it to the right spot, so you're basically silencing the correct gene. So how do we use this for sickle cell? I will preface it saying that they used to use another one called X-gamblogen autosomal, but I'll call it XSL for here on out. But that was only expressing the HbF gene, which expressed fetal hemoglobin. So it expresses fetal hemoglobin so you have more oxygen, however, that was temporary. What they're doing now is moving more towards base editors. But before we get into that, we'll talk about how we actually cure, or what sickle cells is, so that we know how to cure it. So it's a genetic, or it's an autosomal recessive disorder, meaning that mom and dad have to have both of the genes, and that gives the offspring a 25% chance to get the disease. And what happens is cells will start to sickle. And in this picture I have over there, instead of being round red blood cells, they'll turn into crescent shapes or some other misfolded red blood cell. And that prevents how it goes through the vascular system. Additionally, it is a base pair mutation on chromosome 11 that affects P15.5 on exon 6, or codon 6, exon 1 of the HBV gene. And this is what codes for normal hemoglobin. And additionally, this changes the nucleotide codons GAG to GTG, and this changes glutamic acid to valine, and valine is nonpolar. And when this mutation happens, it's the only nonpolar protein in there that'll stick to other valine molecules. So if you see those lines on the sickled red blood cell, that's what causes it. The sickling sticks together and it elongates it. And this causes several problems, especially with pulmonary, cardiovascular, and just especially with your spleen, your liver, and it causes a lot of issues when the cells degrade because they have shorter lifespans, it doesn't carry enough oxygen, and the patient feels it throughout their entire body. So in most cases, these patients can be bedridden. So how we've cured it is with gene therapy. And as I mentioned, they use XSL, but they're moving towards base editing so that it's more permanent because it's only that one nucleotide that needs to be edited. So essentially what happens, they remove the patient's bone marrow, and before they do anything else, they edit it, and then they give them busulfan. Now this is a chemotherapy that gets rid of all of their bone marrow. So this will be a 2-week process, and it'll be really brutal on the patient, but they're gonna monitor them closely so that they don't die, because they're as weak as babies at that point. But while they do that 2-week process, they're editing the genes so that it's correct, and it's the GAG nucleotide sequence. And then afterwards, they inject it back in, and bone marrow can grow on its own, so it's able to grow back. And it takes a few months, but after the first 2 months, they're good to go on their own. So that's how we use it in a good way. But there's another way we can use it, and this is more with epigenome editing. We have regenerative cosmetics. And how we do it, we're basically creating stem cells that make us look youthful and prettier, for lack of a better term. They can do stuff like stimulate collagen and elastin production. It can accelerate tissue repair. It can improve skin texture, tone, and hydration. And it can promote hair follicle regeneration and denser hair growth. And the thing about this, we need to do multiple doses of it as well because these are phylogenetic traits. It's not just one nucleotide, there's several different genes affecting your hair, your skin, everything else about you. So because of that, we need to give them several doses. And again, these are only temporary. So when is it good and when is it not good to use gene therapy or CRISPR-Cas9 for editing genomes? Here I've looked at the philosophy of the image of Dei, which is derived straight from the Bible, and it means the image of God. And the verse for that is in Genesis chapter 1, verse 26 and 27. God says, "Let us make man in our image after our likeness." So God created man in his own image. In the image of God, he created them, male and female, he created them. So this brings up 4 immediate points. First and foremost, humanity and the rest of God is complete. So we have all our genes. God gave us everything we needed. However, we're not perfect. And that brings up 2 more things. Firstly, our genomes are going to be flawed. We're not going to have the correct DNA. That's why we have all of these chronic disorders and genetic disorders. Secondly, we're not perfect ourselves. If we try to add anything God didn't add, then it won't be perfect either. Secondly, the world is a blessing to humanity, so we're taking care of people on it. We also need to take care of the resources on the earth as well. So that includes stewarding it, and I'll talk about that a little later as well. Thirdly, the fact Jesus is fully God, but also just a man, gives us a base model of what our genome should look like. We want to try and edit our genes so it's more like that, and we're not trying to make our own creations. Lastly, the resurrection triumphs over sin, and this includes disease and death. And this gives us a choice. If we pick life, our actions have to reflect life. We can choose to follow God and create super soldiers with our new technology. So how do these two compare? We have genetic disorders and regenerative cosmetics. For chronic illnesses, this is a result of sin, and we're trying to fix it. As Christians, we're going against sin. So fixing that will help us live more better lives and not be bedridden for some of these people. Now, regenerative cosmetics, you're basically serving yourself. However, change is natural. If you're trying to defy age, you're trying to defy the natural order of life, and you're trying to defy how God made you. And that is not something that should be in our ethics. Secondly, gene therapies are safer for the patient. The alternative is just a bone marrow transplant, and these will use HLA typing. Now, if the typing is mismatched, then we're gonna have people that are immunocompromised, or they'll just straight up die. And it's a slow death. I've done research with these people, and it can be brutal. Now, with regenerative cosmetics, you're using these resources. Instead of curing people, you're making your skin tighter. Instead of fixing a disorder, you're making your hair grow denser. And those are not resourceful ways to use such an advanced and expensive technology. Thirdly, we have curing genetic disorders, and this benefits humanity, and not just the patient. It cures your bloodline because it won't be passed down. And secondly, we're just letting people live more meaningful lives. Now, I'm not saying you can lead a less meaningful life if you're bedridden, but if you're a Christian and you want to go out and express your faith and bring others to Christ, you can do so when you're more healthy. Now, if you're just serving yourself with regenerative cosmetics, then you're simply doing something for yourself, and that's temporary at that. Lastly, healing brings life to a patient, and this allows them to have a life that better honors God. Now, Jesus sacrificed for us so that we can pick life. We don't want to disrespect that, so compared to regenerative cosmetics, we're simply fulfilling our own desires. So, to wrap this up, we talked about CRISPR-Cas9. This is a biology that God made himself in bacteria, and we took this good thing and decided to make it better, and we used CRISPR-Cas9 gene therapy and other stuff like that. And that talked about sickle cells and this works for other chronic disorders, not just sickle cells, but this is a good model since it's been researched. We also have genetic editing, which is regenerative cosmetics, which brought up when to use it or not, and we talked about image of deity, which is the image of God. So what would God do in this situation? And I'll leave you with that question. But with that, thank you. All right, any questions so far? Yes?
So do you believe Jesus created disease as a part of the world as like a blessing to humanity, because it allows us to explore different parts of human race?
So disease wasn't intended in God's original design. These were a result of sin. And when you look at it, it's basically like you handed me a vase and then I chipped it. You didn't hand me a chipped vase, I ruined it. That was something we already messed up, but we're not trying to make it any worse with our sins and our transgressions. Yes?
Would you, sorry, I'm trying to find the words here. Is there any form of limitations that should be put in place in order to not let this genetic coding go a bit haywire?
So the only thing that comes to mind immediately is the Geneva Convention, because Germany and a bit of Russia did try this to some capacity, but at the same time, we don't want to make super soldiers. That's not reasonable for the scientists doing that, or the patients themselves. It's just not fair. So assuming we can employ other ethics like that, that'd do the world a lot more good. Yes?
I have a question about the gene therapy for sickle cell. Is that something that's currently running in clinical trials, or are they actually assuming that therapies can improve?
So it just got out of genetic trial or clinical trials as of last year. And I did mention that base pairs and prime editors, those are a little newer, but they've employed those as well instead of XSL. Thank you again for listening, and I hope you learned something.